So thanks very much. So although we've got a little bit behind time, I think you may know, if not, unfortunately our first speaker from Grail in the US is unable to join us. So we have an additional speaker in this session who's Jordi Raymond from the Gustave Roussy, who's got a talk at the end. But also Charlie and Fabrice asked me to talk about the topic of the first speaker as part of my presentation. So I've got two presentations. I'm first going to talk about ctDNA and predicting relapse, and then I'm going to come on to talk about the potential for early detection. And then after me, we've got Alberto Bedelli, who's really been the leader in field of applying ctDNA research, who's going to talk about looking at cancer evolution. And then we'll come on to Jordi Raymond in the end. So I've been asked to come and talk about some of the work we've done in early breast cancer and using ctDNA in that setting. But I've got a little bit of our research we've done in metastatic breast cancer as well. So those are my disclosures. And I'm first going to set just the scene with a couple of slides. So circulating tumor DNA is probably familiar to all of you. But just for a background in case it's not, we know that in particular for patients with metastatic cancer, they release circulating tumor cells into the circulation, but they also release free DNA into the circulation. And this allows you potentially to interrogate the genetics of metastatic breast cancer through analyzing this free DNA that's released into the circulation. This is often very challenging because as well as cancer DNA in the circulation, there is a lot of normal DNA, and frequently, even in patients with metastatic cancer, the amount of DNA that's from the tumor can only be a small fraction of that of the total DNA present. But nevertheless, technological advances allow us to very robustly analyze circulating tumor DNA. And so the question is, can we also use this to try and detect residual disease after patients have been treated for their primary cancer? Now, just to emphasize why this is a problem for breast cancer, and I think this actually fits pretty well after Richard's talk, just to be a bit, to talk about the good sides of precision medicine, although I fantastically enjoyed Richard's talk. This is what's happened to breast cancer in the UK since 1975. This is the incidence of breast cancer in the UK, so it's steadily gone up for a variety of reasons. But over this time, the mortality has substantially fallen, and most of that mortality improvement is through development and application of new treatments. And what that means, of course, is that the majority of ladies treated for primary breast cancer now are cured by their current therapy, and we need to get much better at identifying those women who aren't cured by their primary therapy, so we can focus our treatments on these as opposed to these. And of course, because breast cancer is so common, there is still, unfortunately, a substantial number of women who are diagnosed, treated for their primary breast cancer, but then subsequently relapse and die of it. So to look at this, we looked at our study of women who have treated for primary breast cancer. They all had early stage breast cancer, and they went on to have neoadjuvant chemotherapy and surgery routinely. We had their tumor biopsies, but we also took blood tests to look at circulating tumor DNA at baseline, and then after surgery, after they finished their curative treatment, and then into follow-up. To look at circulating tumor DNA in this setting, you really need to know what you're looking for, because the amount of circulating tumor DNA that's there is very low. So how we did this is we took a amplicon sequencing panel. We sequenced the primary breast cancer to identify the mutations present in that breast cancer. And then we used a technique called digital PCR that very robustly differentiates mutant from wild-type DNA to look for the presence of that particular mutation in the patient's blood samples after they'd finished their primary treatment. Now, digital PCR assays are highly reproducible. This is compared to the sequencing in a Bland-Outman plot, and this is repeats in plasma, showing that you can get robust detection down to below 0.1% allele frequencies. The data I'm going to show you today is in a proof of principle cohort, 55 patients who we treated at the Royal Marsden, all of whom had standard treatment for their primary breast cancer. Now, I'm just first of all going to show you a couple of examples. Two patients who both had the same PIK3CA mutation in their primary. So here is the mutation in their plasma at baseline. This is presentation before any treatment. Here they have chemotherapy and surgery, and in this patient you can see that the mutation's cleared, and then through serial samples in the adjuvant setting it remains cleared, and this lady is now well almost 5 years into follow-up. But here's the next patient with a PIK3CA mutation in their tumour. It's there at roughly the same level in the blood before treatment, but now here after chemotherapy and surgery, the mutation's still detectable. 6 months later, it's increased substantially, and she relapsed 8 months post-surgery. Now, let's have a look at predicting relapse from the different time points. First of all, looking at detection of ctDNA at diagnosis, while the primary breast cancer is still there before treatment. Now, in our study, 70% had mutations detected in their baseline plasma. I'm going to come back to this in the second part of the talk. And those patients who had ctDNA detected didn't have a very different prognosis compared to those who did not have ctDNA detected. Although actually there's strong biological factors that predict whether you can detect the ctDNA. So breast cancer has some very different subtypes. With triple-negative breast cancer, the detection rate was very high, but it was lower with ER-positive breast cancer. And this is intrinsic differences in proliferation between these tumor types. Highly proliferative cancers like triple-negative breast cancer proliferate, but they also have high apoptotic rates, so they presumably release more ctDNA, whereas low proliferation cancers release less, so are harder to detect. But now, if you look at the sample taken after surgery, then this is positive in about 20% of patients in this series, and those patients that had ctDNA detected after their chemotherapy and surgery had a markedly poor prognosis. But I think you can see, if you're used to looking at these curves, that is, that quite a few patients were relapsing late out here that didn't have ctDNA detectable in that one single post-surgical sample. Whereas if you then tracked with repeated samples through the adjuvant setting looking for the ctDNA, then you do in most patients identify ctDNA before they're going to relapse. So you get increased sensitivity by looking for dynamic mutation tracking as opposed to just one sample after surgery. Now, I showed you the difference between ER-positive and ER-negative breast cancer subtypes, and that then reflects on your ability of just a single post-surgical sample. Highly proliferative ER-positive breast cancer, it's easier to predict relapse just with that one sample, whereas in ER-positive breast cancer, potentially you don't need one sample, you do need those serial samples to help give you the sensitivity for predicting relapse. Now, what about lead time over clinical relapse? So this is just one patient who had ctDNA detectable at baseline, nothing post-surgery. This patient actually had a pathological complete response in her primary. So presumably there was very little disease left at this point, so you were unable to detect it. But then over the next couple of years, the ctDNA slowly rises before here she clinically relapses over a year lead time prior to clinical relapse. And for non-specific symptoms at this point, the lady had a scan with nothing seen, and then subsequently she has liver metastases at the point she relapses. And in the whole series, there's a median lead time of 7.9 months between detecting ctDNA and then subsequent clinical relapse. But I showed you this curve earlier, and there were 3 dips here where actually we couldn't detect ctDNA before relapse, nor at relapse. And all these 3 patients had brain-only metastases. So there's clearly some weaknesses to using this to try and identify patients at relapse, that there are some sanctuary sites that tumors just don't release much circulating tumor DNA from. And it's well recognized that for glioblastomas, brain tumors, it is harder to detect circulating tumor DNA in them, potentially because of the blood-brain barrier. But also potentially ovarian cancers release less ctDNA. So again, the abdominal cavity is also another potential sanctuary site where it might be harder to try and anticipate relapse in. Now, we've done longer follow-up than the data I showed you before. Let me just show you back why we were particularly interested in this. Is I think you can see here, there was one patient who had ctDNA detected post-surgery who hadn't relapsed. And that obviously would be a bit of a problem if you were thinking of implementing this in clinic. So we've done a longer follow-up of this series, and unfortunately for this lady, with longer follow-up than we initially reported, she's then relapsed. And so with longer follow-up, we now also can look at overall survival. So if you detect ctDNA post-surgery, these ladies unfortunately have a very poor overall survival. And similar, this is the same result looking for serial mutation tracking. So it's now showing that in this proof of principle series, with 14 patients who relapsed, there was 100% positive predictive value for detecting ctDNA in the adjuvant setting and then going on to relapse. Now, others, of course, are looking at this question. This is another study, and this is not my data, from an Australian group who've collaborated with Bert Vogelstein to look at this in colon cancer. So this is data from 178 patients with stage 2 colon cancer, none of whom had adjuvant radiotherapy. They had a single sample analyzed for ctDNA after their surgery, and these are the patients here in red who had ctDNA detected at that point. And these are clinically low-risk patients, ctDNA also predicting relapse. So this, at least in theory, looks like it has utility across tumor types in identifying patients who are at high risk of relapse. So how we're looking at trying to take this forward is to address the question of, can we use this to guide therapy? So it's no use telling someone they're at risk of relapse and dying unless you can potentially use that information to do something therapeutic for the patient. So we're going to take this forward in a study for ladies with triple-negative breast cancer who've got moderate or high-risk disease and then who've finished their treatment for primary breast cancer, screen them for circulating tumor DNA for their first year in adjuvant follow-up, and then on detecting ctDNA, randomize them to continued standard follow-up, which is observation for these women, or intervention, which will be with pembrolizumab, an immune checkpoint PDL antibody, as a proof of principle in 150 patients to start to address whether we can use this to guide therapy in the adjuvant setting. Now, in the next part of my talk, what we then wanted to go on to try and assess was, after you've identified that there's circulating tumor DNA there coming from residual disease, can you use high-depth sequencing of the plasma DNA to try and interrogate the genetics of the micrometastatic disease? And we did capture panel sequencing with a 273 gene panel to 460x median depth. I just want to show you a couple of anecdotes of what we found here. So this is one patient. These were the mutations found in her primary tumor at diagnosis. and these are the metastasis. I'm showing you this one, of course, because this was one which found the most extreme difference in genetics between the primary and metastasis. Here's the PIK3CA mutation we were tracking to try and detect recurrence, and these are the mutations that are apparently lost, and this is an activating FGFR1 mutation that was gained. Now, this lady had neoadjuvant chemotherapy, so you can sequence the residual disease in the breast after the chemotherapy, and indeed you get reproducible changes, but it doesn't look like the metastasis. But if you sequence the ctDNA here 8 months prior to relapse, the circulating tumor DNA looked much more like the metastasis than it did the original primary. And this is just reproduction of these findings with digital PCR. So at least in theory, you could put this lady on an FGFR inhibitor in the adjuvant setting to try and prevent relapse. Another lady where they acquire a sign SYNE1 mutation, which you can predict prior to relapse, just to show it wasn't just that one patient. But actually, as another patient where the ctDNA didn't look like the relapse, it looked like the primary. So these are mutations that were present in all, and here's an RB1 mutation that was present in the relapse, but not in the circulating tumor DNA prior to recurrence. So we wanted to go back and interrogate this with more detail, so we developed just custom digital PCR assays to look at all of these mutations in the same sample. And then we tracked back all the samples from relapse all the way back. You'll see that this is the same lady I showed you earlier. And so these are the clonal mutations in the tumor which are present here at diagnosis. They go down and then they all come back up again together. And this is the RB1 mutation that's here at the point of relapse, but actually much earlier on in the adjuvant setting, it's not detectable. So this RB1 mutation has increased in clonal dominance in the tumor. And so this is just one of those examples that by the time you have frank metastatic disease, the cancer is becoming very genetically heterogeneous, and potentially one of the advantages of this approach is identifying the cancer where it's much lower bulk to give therapies. And I'll come back to this in just a little while. Now, we have data about this from multiple settings. Here's one data looking at chronic myeloid leukemia, again coming up to this concept that minimal residual disease is a bottleneck, whole genome sequencing in an AML showing that some of the genetic diversity initially there at diagnosis is lost in the bottleneck of residual disease by chemotherapy before then genetic diversity builds again before the tumor relapses. Now, we know this is now very important in breast cancer, and I'm just going to segue a little bit to talk about metastatic breast cancer here, because we know there's one very big, important difference between the genetics of primary breast cancer and recurrence, which is patients, their tumors frequently acquire mutations in the estrogen receptor gene. In fact, they're found in about 20% of metastatic breast cancers, and the mutation clusters in a couple of amino acids in the ligand binding domain where they constitutively activate the estrogen receptor. Now, you can look for these mutations in plasma of patients with advanced cancer. You can use multiple techniques. Here's digital PCR. About roughly half the time when you find a mutation, you find a single one. But in these mutations, you can frequently find multiple mutations. This is a lady with 5 different ESR1 mutations in her plasma. You can also identify our record is finding 8. So these mutations can often be acquired independently in different parts of the cancer. You can show that it's very robust to identify them in plasma, and you only find them in the plasma of ladies with estrogen receptor positive breast cancer. But actually, we're also beginning to show these are important for treatment potentially. This was a study that took patients who had progressed on their first hormone therapy, in particularly an aromatase inhibitor, and then they were randomized between 2 different hormone therapies. This was from a study that was overall was negative. but we went back and we looked at estrogen receptor mutations in the baseline plasma. And the 2 hormone therapies here did the same if you didn't find an estrogen receptor mutation, which was the same result as the overall study. But now if you went back and you looked at the estrogen receptor mutant cancers, they were resistant to this treatment aromatase inhibitor, but they were potentially sensitive to a drug fulvestrant. And this is a drug that degrades estrogen receptor. So you're beginning to show that actually you can potentially target these cancers that have acquired estrogen receptors by using drugs that degrade the estrogen receptor. But where do these estrogen receptors come from? Which cancers get them? Now, you can show there's very specific features that allow a cancer to develop an estrogen receptor mutation. They occur after patients are treated with aromatase inhibitors. So aromatase inhibitors essentially remove estrogen from a postmenopausal lady, and these mutations activate the estrogen receptor in the absence of ligand. So it's kind of obvious that this is the association you find. But if someone's just had tamoxifen, you don't see them. So tamoxifen, this very cheap drug, also at least must have some activity on the estrogen receptor mutations. They occur when patients have responded to their prior hormonal therapy, not if they were intrinsically resistant. And they're also more frequent in bone metastases. And the reason this is that if you look at the original tumor in the primary before it developed an estrogen receptor mutation, you see those that develop them have strong expression of the estrogen receptor and strong expression of progesterone receptor. They're cancers that were initially very sensitive to their hormone therapy, and then they develop mutations as secondary mechanisms of resistance. But the thing that surprised us, and this brings me back to my original part of my talk, is what we were assuming is that aromatase inhibitors are given for years in the adjuvant setting. And I think when we assumed is that that would be the setting in which you would get estrogen receptor mutations, because the lady's been treated for years. But actually, when we went to look at the setting of treatment and when and which ladies got estrogen receptor mutations, to our initial surprise, we saw the opposite. So if ladies were treated with an aromatase inhibitor only in the metastatic setting, they had actually quite a high rate of estrogen receptor mutations in their blood. But actually, if they had their aromatase inhibitor in the adjuvant setting, their rate was much lower. Now, this is not a technical issue of detecting ctDNA, because this is the rate of PI3 kinase mutations in the same samples. So it's not our ability to detect estrogen receptor mutations. But what we hypothesize, and this comes back to my earlier talk, is that in metastatic breast cancer with gross bulk tumor and great diversity in the tumor, somewhere in there you're going to have an estrogen receptor mutation in a cancer cell that's going to grow out over treatment. Whereas in the adjuvant setting, when you're treating micrometastatic disease, actually this is a much more genetically homogeneous setting. And that is, of course, why in this setting, hormone therapies can cure a lot of patients. And likewise, there just doesn't happen to be an estrogen receptor mutant clone there to grow out. So I'm just going to finish this part of the talk with the acknowledgements and acknowledge all the people who've contributed to this research. And what I thought was I'd just take any questions at this point before progressing on to the Grail part of the talk, if you like. So I'll just finish there and see if anyone's got any questions. Thank you. Or not, in which case I can move straight on. Well, as people moving, it's hard to
just
tell if
have
they're moving
a question.
from the microphone. Thank you.
In those triple-negative patients in which you did not find ctDNA after neoadjuvant chemo, did you correlate that with PCR? those are the patients that have PCR or not?
Well, look, I showed you one patient who had a PCR and then had ctDNA detectable and went on to relapse. But, that's obviously more the exception rather than the rule, because obviously PCR is associated with good outcome. And so you see the inverse, which is, patients who have a PCR are obviously therefore less likely to have ctDNA detected after surgery, because they're less likely to have residual disease.
Thank you.
Okay, well, I'll go straight on. So because our visitor from Grail was going to talk about early detection of cancer with circulating tumor DNA, I was asked just to cover this topic. Now, this is none of my research, so you won't be able to quiz me on any of the details of it. But I was asked just to give you a flavor of why people are excited about the potential of ctDNA for screening for cancer. Now, I showed you this slide earlier on, and so this is looking at circulating tumor DNA at the point a lady's diagnosed with breast cancer, and in them, 70% of patients, the ctDNA is detected in their blood. Presumably, it would have been detected, therefore, before that individual patient was diagnosed with breast cancer. And this is the same across multiple tumor types. So this is ctDNA detection in patients with metastatic disease with colorectal, gastroesophageal, pancreas, breast. And here in black is detection in patients with early disease. And I think you can see, although there's a drop-off with this digital PCR assay, you can still detect ctDNA in the majority of patients who presented with early breast cancer. It is related to stage, so there is a steady drop-off in detection of ctDNA as you go from stage 4 to stage 1. But in this series, roughly half patients with stage 1 had ctDNA detectable. But this is looking for known mutations. This is where they found the mutations present in that cancer and looked for that mutation in the blood. And obviously, if you're then going to try and take this concept into screening for cancer, fundamentally that doesn't work because, of course, you don't know what mutations are going to be there to try and identify in advance. But so what are the theoretical benefits? And then we'll come on to the look at the data. So the theoretical benefits, I think, are kind of pretty obvious, really, that if you can detect ctDNA arising cancer prior to symptomatic presentation, then you could potentially detect early disease prior to disease spread and increase the proportion of patients who have curative surgery and potentially reduce those who need adjuvant chemotherapy. And this may be perhaps less important for tumors like Hodgkin's lymphoma that have very high cure rates, but for tumors like lung cancer and pancreas cancer, finding a way to try and identify early stage cancers is utterly critical in improving their chances of cure. So what about the data that you might be able to do this? So this is a case report, if you like, from patients who are having prenatal non-invasive testing. Now, many of you may know about this, but prenatal testing with cell-free DNA is essentially now the standard of care for looking for chromosomal abnormalities in patients, in pregnant women. So the fetus releases DNA into a pregnant woman's blood in the same way that a cancer might do, and approximately 10% of the free DNA comes from the fetus and a mother. And this allows you, through looking for chromosomal imbalances in the maternal free DNA, a way of diagnosing chromosomal imbalances in the fetus. And this is far better than any other test, such as doing a amniocentesis. So it's now entered standard practice. This was a series of 4,000 patients who'd had this cell-free DNA testing for chromosomal aneuploidy, and in 3 of them, they found chromosomal aberrations that looked like they might be cancer. And so the patients went on to have whole-body MRI, and all 3 of these patients had cancer detectable on their whole-body MRI, an ovarian cancer here, a follicular lymphoma here, and a Hodgkin's lymphoma. But by the time you've got chromosomal abnormalities detectable in your cell-free DNA by cancer, you have to have pretty advanced cancer. And I think you might see that this lady with ovarian cancer had stage 4. She had spread of her ovarian cancer. So probably through this approach, you're not diagnosing the cancers early enough. So but actually what you can show is that what you found in the blood does match what was found in the cancer. So this has really spurred on in some part the developments to get very better next generation sequencing done. So the technological advances that have allowed this is predominantly molecular barcoded next generation sequencing. So you take each individual DNA that you're going to sequence and you ligate on adapters that are going to help you sequence that DNA, and these adapters have a unique randomized duplex tag attached to them that potentially in those combinations allow for many millions of different tags. And this allows you to identify that individual single piece of original DNA when then you subsequently sequence it. And why that's important is because next generation sequencing is intrinsically error prone. And what that means is when you sequence this one individual piece of DNA, you get a lot of sequencing errors. You potentially get errors that have arisen because the DNA was damaged by oxidation. But actually, if you can identify the reads that came from this original sequence, that original piece of DNA, the individual mutations will be there in every single sequencing read. So you can cut through the noise of sequencing and get much more exact identification of the mutations. And this technique allows you to cut through the noise. Yellow here is the noise of sequencing by barcoding this molecule. But you're left with some, but then if you then clean up this noise in silico, because you can identify that there are some bases that quite frequently give you mutations, you can actually clean up next generation sequencing to the point where your error rate is very low. And this then starts you to be able to use this technique to look for mutations, very rare mutations in blood without having any knowledge of which mutations you're looking for. And you can start to get sensitivity for one mutation that's pushing down below 0.1% allele fractions. But then if you enlarge your sequencing panel, so you're not potentially just looking for one, you're looking for many potential mutations in the cancer. For example, here, 8 mutations. You then start to push your sensitivity for detecting the mutation down to 0.01%. And at this level of sensitivity, you start to be able to detect really very rare mutations in blood that might be arising from preclinical cancers that have yet to be diagnosed. Now, you can show that these techniques really are very sensitive. So this is some data from Max Dean in Stanford, lady who had chemoradiation for a locally advanced lung cancer, and then detecting here ctDNA at the 0.01% level after finishing treatment before the patient then subsequently relapsed. So the technology is there to do this in theory, but what about the challenges in detecting cancer? And I think this is probably the part of talk the Grail person wouldn't have given you. So I guess this is the disadvantage of not turning up if you're a biotech company. So now let's just talk about the challenges in this. And the challenges are there's probably an awful lot of mutations to detect in us that aren't necessarily coming from cancer. And so what I wanted to show you is some of the emerging data on this, because this is a fabulous idea, but it shows you how challenging it may be to put into practice. So first of all, there's this paper that's a couple of years old from the New England Journal. So what they went back is they went back and looked at a GWAS where they'd done whole exome sequencing on 12,000 people. This was actually a study to look at bipolar disorder and the SNPs that predispose to bipolar disorder. But what they went back and they looked at their sequencing data. Here are the SNPs that they found in the sequencing data. They, of course, center on 50% in the SNPs. But what they noticed is they had this shoulder of mutations that they found here, which actually had much lower allele fractions than you would expect if they were just germline SNPs. And when they went to look in these mutations here, they found that quite a few of these mutations were actually mutations that were known to be cancer drivers in hematological cancers. So what they then went on is to define that actually if you found 3 of these somatic mutations, you cut through the noise. Some of this is going to be sequencing noise. But if you looked for those patients who had 3 or more mutations, you're beginning to get those patients who had evidence of clonal hematopoiesis. And you see that this goes up dramatically with age. So if you're down here, your chances of having clonal hematopoiesis you can detect with these mutations is pretty low. But by the time you're 65, 10% of patients have evidence of clonal hematopoiesis with mutations detected in their blood. Now, these patients who have clonal hematopoiesis had a much higher risk of subsequently going on to get hematological cancer. So you could say this is very useful. You're identifying patients who have a risk of going on to get hematological cancer. This is their survival. There is an increased risk of death, identifying clonal hematopoiesis, but this is mostly because there's an association with smoking. But what I want to draw your attention to here is the scale. So you do identify patients who are at risk of going on to get blood cancer, but only by about 1% a year. So actually, many patients have clonal hematopoiesis with mutations detected in their blood who actually don't go on to get hematological cancer. And actually, the more you look, the more you start to find this. So this is Peter Campbell's data from the Sanger. These are normal eyelids from patients who are having blepharoplasties, where they punched into the eyelid tiny little punches to sequence tiny amounts of the skin of the eyelid in 24 samples from one individual, 92 here. And what you then start to find when you sequence these very small bits of normal skin is actually they contain quite frequent mutations. And these mutations have the same signature of UV light damage as you find in skin cancers. And the mutations you find in normal skin also look like the mutations you find in cancer. So these have been positively enriched in normal tumor cells. And as you sort of map out theoretically 1 square centimeter of your skin, you can actually find that there are clonal expansions with cancer-like driver mutations all over your skin. And I think those of a nervous disposition probably don't want to be thinking about this. But I guess if you're a smoker, this is what your lung looks like. So this just shows that actually there are potentially cancer-like mutations there in all of us. And as you start to look very hard in the blood, you can also find them. This is a paper that sequenced p53 with duplex sequencing to very high depth in a group of patients with ovarian cancer and just control patients who were well. And these are the p53 mutations you could find in some of the control patients in their blood. What do these mean? We, of course, don't know at this point. But the one thing to remember is that actually we've got a blood test for cancer now, which we actually don't do, which is called monoclonal gammopathy of uncertain significance. Now, for the non-medical people in the audience, what this is a monoclonal IgG, immunoglobulin, that you can detect in the blood. And actually, it goes up with age, but actually by the time you're 80, you've got a roughly 5% chance of having this monoclonal protein in your blood that is coming from a clonal expansion of your plasma cells in the blood. We've known about this, of course, for a long time. And indeed, patients who have this monoclonal protein have a substantially elevated risk from going on to get myeloma, but again, roughly at 1% a year. So why don't we screen for this? We don't screen for it because MGUS is a relatively poor predictor of going on to get myeloma, and there is no evidence that early intervention would be beneficial for these patients. So just come to the conclusion. The theoretical benefits of ctDNA screening for cancer really are obvious. If we can detect cancers like lung cancer and pancreas cancer earlier, we could transform the survival for these types of cancer. But really, there's going to be substantial challenges to getting to a test that we can implement in the population. One is going to be the cost of screening. You need to screen big panels to very high depth. But you know, maybe if you can cure patients of cancer, it doesn't actually matter if your test is very expensive, but we might have to invite Richard back on the stage to discuss that. But really, the main challenge is going to be differentiating what I've put inverted commas here is normal clonal expansions that there are in a lot of very well people from early stage cancer. And clearly, this is going to be the challenge that companies like Grail are going to be taking on as they seek to develop this technology. And I'll finish there. Thank you very much. So, look, I'm not going to take any questions on that. If you want to discuss, come and find me at the coffee break. So, look, we'll move straight on now. So we'll move on to Alberto Bardelli from Italy, who's going to talk us about cancer evolution.